We're now going to be moving into our pathology research section. And first off, we're going to be having Dr. Scott Harper, who is an assistant professor in the Department of Plant Pathology at Washington State University and the director of the Clean Plant Center Northwest, located at the IAREC in Prosser. So Scott, if you would share your screen, please.
Okay, good morning, everybody, and thank you for giving me the opportunity to talk about the research that we're doing on the latent viruses of hop. Now, most of this work, I should say, from the outset, is being done by Nin, who's one of my students. So, yeah, most of this work is credited to Nin. So, a little bit of background on the project that we're working on. And hop viruses, as I said on Monday, cause a range of symptoms or diseases on hop. These include leaf yellowing, reduced growth and stunting or reduced vigor, and reduced alpha or beta acid content on the cones. So they affect the productivity and yield of the plant. And once you get a virus, you can't do anything about it except rip out the plant and burn it. There's no really way of treating an infected plant other than to get rid of it. So for that reason, we really try to understand what these viruses are doing. But some of the hop infecting viruses, we don't really know what they do. They exist, they're there, but we don't know what harm or damage that they cause to the plant. And so in virology shorthand, we call them latent, which is pretty much saying they're there and we don't know what they do. In hops, there are two viruses that come under this category, and that's being hop latent virus and American hop latent virus. They're both caloviruses, both aphid transmitted, and both rather prevalent in the US hop industry. Now, these, as I said, these two hop latent viruses, they're very, very common in hop, and they are almost always found in hops. Almost 50, in this case 60% back in 2019 of the hop samples that we'd looked at had one or both of these viruses. So they're very, very common. So what do they do? And that is the point of this project, because in the past, these two viruses have been reported to cause yield losses of up to 13 to 15%, some stunting, shorter nodes, shorter laterals, fewer leaves, and some flecking on one cultivar. However, this is all relatively older research for looking at hop cultivars that aren't widely produced these days, or they're produced in other countries, but not here. So we really don't know what their effect is on US hops. So we have two questions that we're looking at in this study. One is, first and foremost, what do they do on common commercial cultivars? And then, given the damage that they cause, or what damage do we find that they cause, what management is necessary for them? So the approach for this project was, consulting with industry, we found or selected 6 commercial cultivars to study the effects of these viruses, these being Cascade, Chinook, Citra, Zeus, Triumph, and Citra. Now, unsurprisingly, due to COVID-19 related slowdowns in 2020, we were about 6 months behind schedule from where we'd have liked to have been at this point in time. We simply couldn't get into the lab for a while, and it slowed down supplies and other things. But we're proceeding on making more plants, getting everything ready to start the inoculations this year. So in the interim, over the last year, my student has been cleaning up and creating all the plants that we need. So making sure that there's absolutely no viruses or viroids in the starting plant material that we're using. So I started with material from the Clean Plant Center, but checked again and again to be absolutely sure there was nothing there, because we don't want anything to skew the results of the disease assessments. And we've also by doing that, we've changed our approach from our original proposal. Instead of shifting from graft inoculation to aphid inoculation of these two viruses, because we don't want anything else to come along for the ride while we set up the inoculations for these plants. We want a clear, consistent result or infection and result for these experiments. So we're setting up the aphid colony this spring, and we'll be starting to do the inoculations thereafter. So we're creating, as I said in the experimental plan, replicates of 5 different combinations of these viruses by themselves, together, and with a couple of them what we consider more pathogenic viruses, hop mosaic and apple mosaic viruses. Once they're grown, we'll screen them to make sure that there is what we want, and then we'll start assessing the symptoms on these plants. He's created about 100 plantlets of each of these for each of these cultivars by tissue culture. I've got a rack full of these little plants growing up right now, and he's just growing them prior to inoculation. Plants established, colony to be established. But in the interim, while we're waiting to set up this experiment, because this is a long-term experiment, the plants need to grow, be inoculated, and then start growing so we can see the effects of these viruses as they grow and develop over a course of several years. So in the interim, and working with Kayla here on the station, we've been examining the outcome of natural infections of these two viruses on just lines that they had out in the breeding program. Now, these were naturally infected, probably by aphids, because they're very prevalent, and probably been positive for a very, very long time. And what we found when looking at these lines and measuring them by real-time PCR, this is something we do as part of this study anyway, is that there is really quite a differential outcome in terms of infection. In some cases, and this is a log scale graph, so each time that number jumps, that's a 10-fold increase in virus titer. There's a consistent or considerable difference in the ability of one virus or the other, or both, to infect different lines. You can see here a common Chinook over on the end, if you're common commercial lines. The viruses adapt or have adapted to different hosts, or the hosts present more problems for the viruses to infect, depending on their genetic background. And this is something that we suspected, but really didn't have any preliminary evidence for until we started doing this work. The infection outcomes, and therefore the disease outcomes, are going to be different depending on the cultivar. So this, in a way, is a justification of what we're trying to do with this study, to try and figure out what are the outcomes for infection and therefore for disease. So we hope that what we learn from this project as it progresses was that we'll be able to assess the risk of these two viruses, these two latent viruses, on hop production, and therefore determine what needs to be done in terms of managing them. This will also aid in developing, in the long term, new virus-resistant cultivars, because the hop breeding program can use this information to screen different lines as they're developed. So thank you for your time, and I'd like to thank the Hop Research Council and Washington Hop Commission for supporting this project, the Hop Breeding Company for providing the Citral line, and the National Clean Plant Network for providing much of the clean plant material we started with, as well as some of the facilities that we use. Thank you. Any questions?
I'll check the box here to see if any questions have been submitted. And if not, again, if questions come in late as the other speakers are presenting, we can certainly circle back to you, Scott, as they come in.
Okay. Thank you, everybody.
Okay, thank you, Scott. So next up, we're going to be having Dr. Dave Gent. He is our research plant pathologist at the USDA-ARS in Corvallis. And joining him are a number of his staff and graduate students, including Mary Block, Teddy Borland, Briana Claassen, and Michele Wiseman. So Dave, if you're ready, I'll hand it over to you.
Great, I am. Thanks, Judy. And I'm going to present just a few slides, and this is very much going to be a high-level overview. And what I'd like to do is not repeat the work that's been presented in the recorded presentations, but just try to situate and give folks an understanding of the work that we're doing at a high level and how it fits in with overall HRC priorities, introduce a few folks in our group, and then spend most of the time just trying to answer questions today. And so what you're looking at in this screen on the left, I took the HRC research priorities document from last year, and I made a word cloud out of this. And so if you aren't familiar with how word clouds work, the most prominent word that appears in text appears in the largest font in the middle. So the things that the HRC, their priorities relate to management mostly. And that fits well with what we do. We are ultimately interested in how do we translate fundamental and applied work into improved management for the growers. If you look at the work that we're doing, it fits broadly under the HRC priority areas of plant health. If you looked at my talk, you might be surprised to see that we do some agronomy work too. And we do some work in arthropod pest management. So the reason this is so diverse is because growers are managing not one thing, they aren't siloed, they're looking at everything in the whole system. And so we try to situate our work in the overall production context as well. There's two projects, broad projects, the HRC funds in our group. The first is this one at the top here, and this is mostly focused on more fundamental and long-term work on basic disease biology and ecology. And here's another word cloud I made. And so in this project, what you'll be hearing, what you heard from Teddy Borland was some of her work related to Fusarium canker. So Sambucinum appears prominently in this word cloud. That's the species name of the Fusarium canker fungus. Also, some of the fertility-related work with nitrogen sulfur appears in this project as well. And the second project, this is this one, and this is work that's more focused on immediate and short-term and intermediate-term needs related to prominently the powdery mildew and downy mildew. And we're focused largely now on maintaining EU market access, mitigating some of the potential issues that are developing in the EU and the regulatory front, and ensuring that growers have best practices and tools for managing the priority diseases on varieties they already have in the ground and they're producing now. This is the team in our group that makes this happen. And so you'll hear from 5 of these folks today. And I'll give just a high-level overview of this work. So Briana Claassen, what you heard from Briana, Briana's a faculty research assistant in our group. And so Briana presented work in her presentation on the overwintering biology and management of hop downy mildew. Teddy Borland is a PhD student in our program focused on Fusarium canker. And so you heard from Teddy's presentation, working the biology, ecology, and management of this increasingly important disease. Mary Block is a former master's student who's now a faculty research assistant. And Mary presented on novel assays for detecting certain pathogenic races of the fungus, and then also some characterization of pathogenic diversity in the organism. And then Michele Wiseman, formerly a senior research associate in our group and now a PhD student, who's breaking some new ground on a novel form of disease resistance in hop. So what I'll do is I want to just give just a couple bullet points of some of the accomplishments that you saw in this work. So in the work that Briana presented, one of the major accomplishments is new knowledge on the ecology of downy mildew and the value of post-harvest fungicide applications and management. This is a question that I get a lot from growers on, do I need to be doing anything after harvest for downy mildew? Briana has some work that informs that. Briana looked at fungicide programs, how to maximize the effectiveness of currently registered crop protection tools. And her work also identified 2 new modes of action for downy mildew that are now in the registration pipeline. Teddy's work, what you saw is that she is involved with development of a diagnostic assay for detecting and quantifying the Fusarium canker pathogen in environmental samples. This opens up a lot of new doors in terms of as a research tool, but also for risk assessment as well. Teddy's work presented some of the first data on validation of various cultural and chemical control measures for canker, and more fundamentally, knowledge of the pathogen host range and genomics. In Mary's work, what you saw is that there was an assay, a DNA-based assay where you can rapidly detect and determine whether a given powdery mildew on a plant, whether it's capable of infecting plants such as nugget that have the R6-based resistance. So this is very useful for us as a research tool, but it's also some practical implication for risk assessment. And what you also heard from Mary is work on characterizing the powdery mildew resistance in the variety strata as well. And rounding this out, Michele in her presentation spoke to identification of a gene family in hop that's associated with powdery mildew susceptibility. So it's the opposite of resistance genes that confer susceptibility to powdery mildew, and identifying possible targets for breeding that we think would be more durable and long-lasting forms of resistance. There's a other things we're working on, and I won't bore you with those today, but just know that not everything that HRC was supporting made it into presentations just because of the constraints of time. But if you are stuck at home with nothing to do, or you have insomnia, there's not one but 2 HRC reports where we document the progress for the year. And with that, I'm going to just thank you all for being here. Thank the HRC for your support. And I'd like to turn this time over to the audience and spend most of it answering your questions. Okay, so with that, Michele, Palacios, if there's any questions from the audience, I'd be happy to try and answer them. And our team as well, that's online.
Oh, okay. Great. Let's see. I have a little lag on the questions, so I'm turning it over too quickly, I realize, because I'm always getting some in right at the end. Let's see. We did get one in from Grayson. I don't know if I'm able to pronounce some of these. Thoughts on Trichoderma or mycorrhiza on fusarium? I didn't get that second. You might need to look at it to interpret what from my pronunciation.
Sure. So, Teddy, would you feel comfortable fielding that one?
I can, yeah. So we haven't looked into the use of like Trichoderma. So that would be fungi that feast on other fungi, basically. And so using that as a management method, which I think this question is relating to, we haven't looked into that on hop. And we haven't really looked into mycorrhiza in relation to fusarium on hop either. So.
Maybe one thing I'd add to that, more broadly, there is some work, there's very little work on fusarium canker and hop in general. So the data that Teddy's generating, our group's generating, some of the first. There is, in other systems, there are soils that can be more suppressive to certain fusarium diseases. Probably several mechanisms associated with that.
All right, another question. What base varieties are you all looking at for base breeding on the powdery mildew durability piece?
Sure, I'll bounce that to Michele Wiseman.
Yeah, that's fine. So initially what we're trying to do is just identify the genes. And so kind of by essentially comparing varieties that are susceptible versus resistance, we can do some gene expression studies and identify the differentially expressed genes. And once we have a good idea of those genes through characterization studies and functional studies, then I believe there's another collaborative project going on. But we're like a smaller part in that, but some of the varieties they mentioned were Nugget. Dave, do you know the other ones? I know that was the first one I remember.
Yeah, they were all ones that possessed ARS as the starting point in that project, Michele.
But to answer the question, we haven't really identified varieties long-term for the MLO project. So that's going to be a few years out. But of course, we're open to feedback. Yeah.
I think what we'd expect, so Michele mentioned MLO. MLO is a gene family that is involved in several functions in plants, not just hops, but in monocots and dicots. And its lack of function mutations in certain MLOs that can confer powdery mildew, lack of susceptibility. So it's probably present with Michele has evidence that it's present in even very susceptible varieties, like some of the high alpha varieties you find MLO expressed. So, but what we're looking for is not the presence of the gene, it's the absence of the gene or non-functional version of it. So it might be that we find this broadly distributed in germplasm, Diane.
Yeah, and so in that case, I guess we'd be like doing more, probably more QTL mapping with the extant germplasm in order to develop molecular markers for broad use in public and private breeding. That's kind of the end goal.
Another question from an attendee is how can growers get involved in research opportunities or collaboration opportunities? And is there any research into environmental sustainability?
Sure. So, so maybe this is for Scott and for our group. It's all for our group. So we do a lot with growers because it takes a lot of real estate and a lot of resources, and there's certain things that we just need to scale, and we want to have in a commercial field so it's well situated. So if you're just contact me. I'm easy to get a hold of. Google my name or email me, whatever, and we're happy to. We do a lot with growers. You know, be careful what you ask for because we can be a little bit demanding and difficult at times too. But yeah, we're happy to work with folks. And I guess the second part of that was on things on environmental sustainability. You know, we are. I'm hoping everything we do feeds into that goal. I mean, we're working towards trying to reduce inputs wherever we can. It's difficult. Some of these are long-term kind of recalcitrant problems, so progress tends to be slow, but I hope it's all working that way. There is a broader effort underway with Doug is leading and others that are on this webinar involved with for doing some benchmarking work. That's proposed. So, but I would hope that everything that we're doing in some ways contributing to the environmental sustainability goals.
I'll just comment on that environmental sustainability goal too, is that, you know, a lot of the research that John is doing and that we are doing, looking for molecular markers for powdery mildew resistance, is kind of with the long-term goal of putting in less input, so applying less pesticides, and you know, therefore kind of having more of a less of an environmental impact with those synthetic inputs. So kind of that's the long-term goal. I actually have a question for Scott. Can we ask? Oh wait, somebody else asked a question.
Oh yeah, we did get another question come in. Yeah, we need to circle back to Scott as well. But before we'll do Scott's question, let's go ahead and answer from Benjamin. Do you track the emergence of new powdery mildew races across global production regions that might pose a threat to existing sources of resistance?
I try to stay abreast of what the Europeans are doing in terms of usually you're looking for a needle in a haystack here. So in terms of trying to actively screen powdery mildew isolates in the population, what you're looking for is these very rare mutations that lead to virulence, and it's basically impossible to find those at a low level. Usually what happens is when a given source of resistance is deployed broadly, you'll find disease developing on that after some amount of time. It might be a few years, it might be decades later. And so it's really difficult to proactively detect those. So there is screening that happens. So we try to be aware of what's in the population, and then we do screening of, if we see something that's unusual. There's some fundamental limitations on, I mean, you can't really do more than, you're talking about like tens of isolates a year. Mary does this, she could speak to how much effort it is to do this. So really it's not proactive, it's usually after something emerges is that when we can actually detect it practically.
So Michele, you had a question for Scott. So maybe let's catch back up. Scott, you had the question come in from Ryan, is when and how do you plan to assess plant damage caused by viruses?
Good question, because we don't really know when in the life cycle or when in the seasonal cycle these viruses will, or where on the plant these will cause damage. So we're going to be monitoring them as they grow, looking at their growth rates versus the existing, against negative controls, of course, and other viruses. We're going to be trying to monitor their photosynthetic activity, because often some of these viruses and ones from this particular family will cause damage to chlorophyll, chloroplasts, and may slow down their production, so manage that with the glycol system. And then when they finally start to reach bearing age, we'll be assessing the cone content to see if there are any biochemical changes, alpha and beta acids specifically, to see if they are causing anything that really affects the product coming from these plants. So kind of a broad question, broad answer. We don't know what sort of damage they're causing, so we're just going to have to try and look at the obvious potential things that could be happening to the plant and see what happens.
Thanks, Scott. Michele, did you want to ask your question of Scott before?
Yeah, so I was just curious, Scott, if you've done any if you looked into the range and transmission rate of the hop latent viroid between hemp and hop. I know that's a big concern with hemp growers.
No, because, well, but for some federal reasons, but it's no, it's for hop latent, the viroid in particular, which again, not the focus of this particular study, but very common and something we do look at on the hop side for clean plants, particularly. It's mechanically transmissible. We don't know of any other, potentially seed, but that we haven't conclusively proven. But you would have to transfer it via sap or in fact, on cutting tools. Something can transfer sap from plant to plant via a wound to get it from, particularly across those kind of species barriers. So we're not really sure how it happens or how it got there. I suspect that in hops and in hop relatives, I have to tread and word this very carefully considering my funding agencies, that they've always been there. And most of the time do nothing. And they're very selective in the damage that they cause, if at all. So in most hop lines, hop latent viroid does nothing, absolutely nothing, that we're aware of, because we haven't got a hop line without it to know the difference. So it's an open question at this time.
Thanks, Scott. A question from Ryan. Which powdery mildew race would you suggest for using in a hop breeding program screening?
Let's see. Mary, do you want to field that one? You're the one person in our group that handles powdery mildew in the lab and race characterization.
Sure. So I would suggest, if we're doing screenings in the Pacific Northwest region, the predominant races that we see here are the pre-2012 race, which is characterized by infecting hop cultivars that have the RB, R3, and R5 base resistance. We also have a powdery mildew race that's able to infect hops that have the partial resistance to powdery mildew, such as Cascade. And then the third predominant race is the V6 powdery mildew that can cause disease on hop cultivars with R6 base resistance. So with that in mind, we have these three predominant races, and to be really thorough, I would suggest using all three races when you're performing screening, because if you use just one, there could be potential to be missing some other form of resistance that we have out there. So that would be my suggestion, to do a series of screenings with the three different predominant races that we have here.
Yeah, that's right. It might depend a little bit on the breeding objectives and where you're located too, Ryan. And if there are races in Europe, and we have these in our collection that can overcome like R1 and R2. So it depending on the nature of resistance you're breeding for, the strains you use and the sequence that you use in the inoculations varies somewhat, but Mary gave for the Pacific Northwest, that covers the known pathogenic diversity at this time.
A couple other questions. Has the new understanding of downy mildew led to new management practices? From Julian.
I can speak to that. So from the biology standpoint, we found that if you reduce infection earlier in the spring and summer, that can reduce overwintering to the next year. And specifically relating to management, the spray program studies we conducted, they suggest if you use more effective chemistries or in the earlier sprays in your spray program, that'll lead to better disease control later on in the season.
Okay, let's see. Another question has from an attendee, has there been any research done to identify if there are differences in disease susceptibility between diploid and polyploid varieties?
Not systematically, to my knowledge. Someone like John or Kayla could speak to polyploid better than me, but generally you see an increased size of plant organs. I mean, like if you take Millennium and Nugget, for example, just larger leaves, kind of larger, more vigorous plants. And so, whereas both Millennium and Nugget both have R6, there might be some indirect effects due to, if you have like just a larger canopy structure, that all other things being equal, that would tend to potentially lead to greater susceptibility, or at least more difficulty managing, say, powdery mildew. But yeah, I don't think it's been looked at systematically, though, is the short answer.
All right, another question from an attendee. Have you done any work on soil health, i.e. the beneficial microbes in the biome and their effect on potentially mitigating disease?
Allie, who's on this webinar, that would be in her wheelhouse. So my group hasn't went there yet. So I'll defer that to her for later.
Okay, looks like we've got a pause in questions unless I missed something. Oh, I've got, let's see, a question came in for Teddy. Your study on Fusarium canker showed 2 drip lines, showed a higher density of wilted vines. Was the water application rate higher in total for the 2 drip lines? Any thoughts on what caused this result?
Hi. So yes, it ended up being that the amount of water that was coming out in the double drip line system was higher than in the single drip line system, and we do think that that may have something to do with it. Overall, disease levels were very, very low this year. So in that presentation, you can see there were really only a couple of wilted vines overall. So it's hard to draw any major conclusions from that. But for the next growing season, we're evening out the amount of water that's coming out of the 2 systems.
A question from Julian. Is there a list somewhere that rates the overall effectiveness on downy so that we can use most effective sprays first? Generally, we start with tank mixes, including copper, so that we don't end up with copper residues later in the season. Would you recommend pushing copper in favor of systemic controls early in the season? And then he also offered his email, so you can see it in the, if it's easier to continue the conversation via email for panelists to see.
Yeah, so in general, if you want to get the rates that we used and then the different products and how they rated against each other, that could be found in the HRC reports. I'm not sure which one though. It's not the non-fusarium one.
Correct.
So, and then the second part of the question, I'm going to pass that to Dave about pushing the copper.
Yeah, so Julian, what we did in Briana's work is that specifically it was the Curzate or cymoxanil and copper in the first two applications of the season seemed to give enhanced control. So I don't think that your strategy of using copper firstly early in the season is necessarily inappropriate. I think it would be important to have something like cymoxanil or another fungicide that has strong post-infection activity in your first two treatments of the season. And so we've seen this pretty consistently. So that's an old Curzate copper, as if, I mean, this is old chemistry, but it seems to fit very well in the early portion of the program. So you had a question there about, is there a list of overall effectiveness? What we are doing, this will probably take another year or longer to finish, but someone in my group, Steve Massey, has digitized all the historical records from the public downy mildew fungicide efficacy trials, and we're intending to do a meta-analysis of, it turns out to be about 20 years of trial data. So we're hoping to get this a cogent, data-driven comparison, but that's in process. But as of right now, though, Curzate copper looks like it's a good option in the early portion, like the first sprays of the season.
How are we doing, Michele? Do we have any more questions for Dave and for Scott?
I'm gonna check all the boxes here. Right now we are caught up on questions, it looks like.
Okay.
Is there, I just wanted to clarify, are we, the presentation from Dave, is there an additional presentation or are we waiting for just more questions for this session?
No, this was it for Dave's group. You know, he's got a big group, and so we wanted to give them enough time to answer all the questions. Plus, it looks like we've got 181 attendees here.
Okay.
So the other thing is we're running a little bit ahead, which is fine. So if people come up with questions, we can always just push them off into a different session and then redirect them to Dave and his group.
I do have some more coming in though. Yeah.
Okay.
Yeah, that's why we have to fill time as people type. So a question for Mary. Your research found that the varieties strata can develop powdery mildew when challenged with the pathogen strain from Cascade, but not Nugget. As acreage of strata is increasing in Oregon, what are the potential implications of your finding for producers of Cascade and Nugget or other varieties with similar powdery mildew resistance?
Yeah, so I can address that question. So based on the preliminary studies that I performed in the lab, it appears that strata has a partial resistance to powdery mildew similar to that found on Cascade. So something to keep in mind is that as more strata yards are being planted, you have to think about what the landscape is out there in the surrounding hop yards. If you're planting strata yards to a region that predominantly has some older Cascade yards that are known to have high levels of powdery mildew in them, you may want to be really mindful of taking some preemptive action, scouting those strata yards because the powdery mildew can transfer between strata and Cascade and vice versa. When looking at Nugget, that has the R6 based resistance. And what I found based on these early studies is that the V6 powdery mildew that infects Nugget isn't as easily transferable to strata. So there may be a less risk of having strata yards planted near a Nugget yard. However, within Oregon, we are seeing a more nugget yards being removed. So what I'm thinking that we may be seeing in the future is, as more of those yards are being removed that have the R6 base resistance, there could be a change in the overall powdery mildew population here in this region. So we may be seeing a move from more of the V6 powdery mildew to a powdery mildew that can infect varieties that have the partial resistance to powdery mildew. So that's just something to keep in mind.
Okay. We have a question for Briana. Your research suggests that infection by the downy mildew pathogen earlier in the season, spring and early summer, rather than infection in autumn leads to, or autumn tends to lead to greater levels of disease next year. Why might this be?
Yeah, so when the pathogen infects earlier, we think that this gives the pathogen more time to infect deeper into the crown and then have more chance of survival in the developing buds that are going to come up in the next season. So.
Okay, thanks. So I want to check our question box. Let's see, looks like we have, this question would be for Michele. What is durable resistance? Is it possible to know if the powdery mildew resistance in a variety will be durable before that variety is released?
So durable resistance is broadly defined as essentially a plant that's resistance has remained effective over a period of time in a that's been widely cultivated in a place that favors disease. So it's kind of a broad definition, but essentially just resistance that remains in light of the presence of the pathogen. And then can we know whether it will be durable before it's diploid? Well, we can try and test it, you know, under a greenhouse and in field trials against the known races in the region to test for durability. But unfortunately, to be honest, anything that's widely diploid over a long period of time, if there's a lot of disease pressure in that region, kind of like what we've seen with Nugget and Cascade, it's hard to know how durable the resistance will be. But one way to make something have, you know, select for more durable resistance is to, well, you should check out my talk, is one of the newer strategies is to target susceptibility genes. And that kind of prevents a pathogen from even infecting in the first place. And so that's been proven to be very durable. So yeah, just looking at strategies that they use in other crops, like in my talk, you'll see the durable resistance they've gained in barley by targeting the mildew locus O susceptibility gene. They've had those varieties deployed for, I believe, like 50 years now, and they're still resistant to powdery mildew. But that's kind of a long answer for no, you can't always know, but what we can do is follow other crop models to get a good idea, and we can do smaller trials in the greenhouse and field trials to get an understanding of the durability of the resistance. Dave, do you want to add anything to that?
No, I think you nailed it, Michele. It's kind of by definition, you have to have something broadly deployed for a long period of time to know if it's durable. But I guess we're just like what you said, we're relying on examples of what's worked in other systems, and that's really the best strategy we can propose at this point.
Looks like we have another question for Teddy. We're relating to that double drip tube again. And the question is, in your presentation, you indicated that the double drip tube configuration for irrigation increased fusarium canker. Did you expect this, and what might explain this response?
Yeah, so originally the idea was that spreading out kind of the distribution of water around the hill versus having the drip line directly on the base of the vines may reduce incidence of fusarium canker. But as you mentioned, we did see a little bit higher density of wilted vines. And that kind of circles back to the last question I answered, where we are thinking it could have had to do with the fact that there was more water overall coming out of the double drip tube system than the single drip tube system. So yeah, again, we're going to be continuing it this year and correcting for that water differential.
A question for Michele. Are there potential issues that might arise from using MLO-based resistance?
Yeah, that's a good question. Unfortunately, MLO-based resistance isn't a silver bullet. Oftentimes when we target MLO in the form of a loss susceptibility gene mutation, we see other effects such as like what are known as pleiotropic effects. That just means there are other phenotypic changes that often occur. So, you might see yellowing of the leaves, or you might see stunting or something like that. So they've observed these pleiotropic effects in a number of different hosts, such as Arabidopsis, in barley, and I believe in apple too. That being said, there have been numerous hosts where they haven't had those pleiotropic effects. So that, remains to be known in hop. Another thing that could happen is there have been some studies that have indicated that loss of function MLO mutations can lead to increased susceptibility to other pathogens, and these are usually necrotrophic pathogens, so pathogens that feed on dead tissue. Well, opportunist pathogens. Once again, that's not always true, so we have to look into it. Of course, my dog wants to play as soon as I'm talking. And then the last thing that could be a negative effect is it could have an effect on the colonization rate of arbuscular mycorrhizal fungi. And those are really highly beneficial to plants in a number of different ways. So we don't want to decrease the rate of arbuscular mycorrhizal colonization. If it has a net increase effect, that's wonderful. But there have been a few papers that have cited a net decrease, and then others that have cited a net increase. So yeah, unfortunately, MLO, there are a lot of different factors we need to look at before deploying a variety with an MLO-induced resistance mutation. So loss of susceptibility mutation, which leads to resistance. But it has been documented to be successful in a number of different hosts. So my hopes remain high, but it will be a long process, and I'm sure you all will hear about it for a few years at least.
Thanks, Michele. A question from Mark. Are there species in the areas surrounding the hop yard that should be mowed or tilled because they harbor powdery mildew and other fungal pathogens? In particular, we have goldenrod near us.
Yeah, good question, Mark. Typically, powdery mildews are very restricted in their host range. So they tend to affect plants in the same family, sometimes the same genus, sometimes just individual species. So for example, like the apple powdery mildew fungus goes to apple and not to hop, and vice versa. So the goldenrod, I mean, maybe if you see mildew on its indication that it's indication of generally favorable weather for powdery mildew, but it's not a risk in and of itself. There's one of the posters, Michele's poster has some early data on an alternate host for the hot powdery mildew fungus, which is hemp. So I guess you want to mow down the neighbor's hemp potentially, but but that's all that's all early data. But that's the only other plant that we know of that could be an alternate host for the hot powdery mildew fungus at this time.
There was a follow-up about what about breeding with MLO-based resistance and potential linkage drag or effects of genetic backgrounds?
Could you repeat that
Yes.
question, please? Or I'm sorry, Michelle, maybe you heard it.
Yeah, I can repeat it. Or yeah, if, it's in the chat, so I don't know if you can see it as well. But a follow-up was with that MLO was, what about breeding with MLO-based resistance and potential linkage drag or effects of genetic backgrounds?
So I would say I'm going to be very cautious with this answer. This is something that we don't know, and unfortunately has not been widely documented in other hosts. I think there's always a potential for linkage drag, and this is just something that we're going to have to explore in order to really know. Dave, do you have any?
You know, I'd be more concerned about the pleiotropic effects you just spoke to than linkage drag. At this point, the idea of linkage drag, that's probably the case with any, it's not just limited to MLO, that'd be any R gene potentially. This is, it gives us another target, and this is early stages, so we'll see as this develops, and yeah, that's the best answer I can give at this point.
All right, a question from Kayla. In cases where you're selecting against powdery mildew susceptibility, what sorts of underlying mechanisms or morphologies do you hypothesize are contributing to susceptibility? You want me to repeat it? Did you catch it all? I've got a train coming through now, of course. Welcome to Hubbard.
So,
repeat it?
well, I think I broadly understand the question. So you're asking like kind of what phenotypic, I think the kind of the main question is what kind of phenotypic effects are you expecting to see with a loss of function susceptibility mutation? Is that kind of, I think that's kind of what I'm getting at. Dave, is that how you understood that question?
Yeah, I think that's a good way to answer it.
Okay, all right, yeah, so with ML, at least if we're looking, so we don't even need to talk about specifically with MLO. When we're trying to think about loss of susceptibility gene mutations, we're trying to think about, you know, what is required for a pathogen to cause disease in a host. So with these loss of susceptibility mutations, the pathogens are interacting with different factors in the host that are required to cause disease. So, for example, with MLO, the pathogen is, you know, oftentimes the hostorium, so that's a part of the powdery mildew fungus, penetrates, but then it can't actually cause a host reaction because there's, you know, some sort of mutation within that process that doesn't cause a response. So it's what we're looking for is some sort of interruption within that kind of host microbe interaction system that doesn't cause a host response to the pathogen. Because oftentimes it's like this hyper sensitive response that causes issues. Dave, do you want to add anything there?
So I think that maybe if I was to answer that question more broadly,
Doing it too molecular.
Well, just to add on to what you said, so I guess MLO is one form of like a susceptibility gene. So you wouldn't necessarily see anything morphologically different in an MLO plant, assuming there's not some pleiotropic effect. So there could be other forms of, like more broadly, in like lack of susceptibility. There is an association potentially with cuticular thickness. So that might be something that a morphology or a phenotype that could be associated with this. The time, there's a period of juvenile susceptibility. I'm speaking of hops specifically, but this is broadly true of powdery mildews. There's a period of juvenile susceptibility in the leaves, and as the leaves age, they naturally become more resistant. And the process, like how long they're susceptible, varies between varieties. So some work that Michele did and our group has done looked at this, like in a cascade, for example. So even with isolates that are very long cascade, it's a briefer period of juvenile susceptibility in Cascade versus something like high alpha, highly susceptible individual. So that, I'm not sure the morphological characteristics associated with that, but I think that it's related to, I think it's reticular thickness in part. So Kayla,
just to add on to what Dave said, is just that oftentimes you can't see the morphological or any kind of phenotypic macro changes. So that's why we're looking at the genetic level. You know, the obvious phenotypic change would be resistance. But to know that it's recessively inherited loss of susceptibility, susceptibility, loss of function, mutation, caused resistance, you don't really know unless you do some gene expression studies or some other functional studies.
I think, Kayla, one other, thinking of like disease avoidance or clindusity. So is there some general plant architecture or other characteristics that make mildew harder to control? Canopy density in general is associated with how hard powdery mildews are to control. And so a lot of the cultural practices that we advocate to growers are all about minimizing canopy density. So if you have a plant that produced a greater ratio of cones to leaves, I would expect that would be a morphological characteristic that would make powdery mildew maybe not less susceptible, easier to control, certainly.
Hey, Michele, I think we have time for one more question, and then we'll switch over to the next section.
Okay, great. We've still got questions coming in, so we'll squeeze this one in then. From Blake, were there any additional known varieties that lost resistance to powdery mildew in 2020?
Not that I've been alerted to. Comet has a largely uncharacterized form of resistance to powdery mildew. And I think that in the early work that we did in the lab, it looked like it was partial resistance. Like if you could put enough mildew on Comet, you get a small amount of disease. There was reports of disease on Comet. I think that was in 2019, Blake, not 20. I'm not sure that indicates the resistance in Comet it's been overcome, or it's just a reflection of extreme disease pressure.
I'll just address Megan's question real quick.
Okay.
So Megan asked, Michele, can you speak more to the powdery mildew host range indications between the Canabaceae species? Are we seeing new species of powdery mildew that can go from hemp to hop and vice versa? What about powdery mildew infecting hemp? Briefly, this is all summarized in the poster, and I just want to say that this is all very preliminary data, but the only species that we found in the field on both hemp and hop is Potosphaera macularis. Now, we have seen Golovinomyces ambrosiae, which is the one that more commonly occurs on hemp, in like glasshouses and indoor production on hemp. However, we've only seen Golovinomyces ambrosiae on hemp in the field and not on hop. So when we've done, when Bill Weldon did inoculations in the lab, and when I've done detached leaf studies in the lab, I found that hop remains a poor host for Golovinomyces ambrosiae. So I'm not at all worried about Golovinomyces transferring between hop and hemp. I'm much more worried about Potosphaera macularis, because Potosphaera macularis can cause pretty severe disease on both hosts.
Hey,
Quick timing. Yeah, that can.
Yeah.
We've got them all answered. So.
Okay, great. And if you have any other questions too, you can always put them in the Q&A, and either Michele or I could put them in the chat and send them to the panelists. So thank you very much for your time. Excellent questions, excellent answers. And I would also encourage all of the attendees to go and see the full presentations. I actually viewed them in the morning, Dave, with coffee. as well as the posters. And remember, we're going to have the poster blast this afternoon.